For those of you sitting and waiting, we're just giving 1 or 2 more minutes for people to trickle in. So I know the silence can be deafening. All right, well, let's just go ahead and get started. Welcome to the 2022 State of the Medical Device Industry webinar. We're going to be discussing some different changes to quality, biocomp, and some of the other changing regulations. When we planned this webinar, we were kind of thinking about focusing on EUA or a high-level overview, and then possibly, a deep dive into biocompatibility. And that's still the case. We are going to touch on some of the new QSR to QMSR, the proposed ruling from the FDA as well. So just an FYI to let you know what we're going to be talking about today. My name is Etienne Nichols. I'm a medical device guru here at Greenlight Guru. I'm a medical device engineer, started in manufacturing, product development, went into project management, and now I work with Greenlight Guru, helping customers get to market faster, more efficiently to get their devices safe and effective to market. So I'll go ahead and let Thor introduce himself.
Thank you very much. Thor Rollins. I'm a senior director at our kind of biocompatibility / toxicology group here at Nelson Laboratories. I have been working in the biocompatibility industry for 20 years. I am an active member of the ISO 10993 TC194 working group that writes the ISO 10993 standards, and I absolutely love biocompatibility. So I tend to talk a lot about it, and I'm excited to be here today to kind of talk about some new things that are happening within the ISO TC194.
Awesome, and I appreciate getting to be here with you as well, Thor. So let's go ahead and get started. So on our agenda, as I mentioned, we're going to be talking about some of the transition away from EUA, emergency use authorizations, as well as the breaking news from the FDA about their proposed QMSR. That's supposed to be QMSR. I'll change that slide later. But just where we've been and where we're going. And then as Thor mentioned, we'll be talking about the ISO 10993 updates. That's going to be a much deeper dive in the details as to that change. We'll be doing a high level overview of the first two. Now, if you have any questions, go ahead and put those in the chat and we will answer those at the end. If we catch them as we go, we'll definitely try to catch them that way, but put them in the chat and we'll get to them at the end if not. Okay, so to start out, the transition from EUA. So we'll talk a little bit about some of the history first. As some of you know, if you've been involved in an EUA or a device that is marketed under an EUA, the public health emergency was declared by the Human Health Services on January 31st, 2020. Since then, over 900 EUAs have been issued. Now that's a low number. It's when you look at the FDA website, it may be closer to 1900, but 900 is kind of the official number put out. What are these devices? So these are unapproved medical products or unapproved uses of approved medical products to be used in an emergency to diagnose, treat, or prevent serious or life-threatening diseases or conditions caused by CBRN threat agents when certain criteria are met, including where there are no adequate approved or available alternatives. So the coronavirus pandemic is what this stemmed from. And we've been going along for, we've been in the pandemic for a while. Most of us, maybe we're a little bit used to it at this point, which is, but it may be coming to an end. So the FDA put out a proposed transition plan for the EUA in December. And so some of the things that are involved in that transition plan are the following. So the FDA does not intend to require market removal from the following categories. So these are some of the categories that they've talked through. Now, I do want to mention this is a proposed rule. So there is a comment period. The next slide, I'll show you a little bit more about that. But let's talk about through some of these. So if you are a single-use, non-life supporting or non-life sustaining device, and that is going to be consumed by the end user, the FDA is not intending to require that marker removal for that category. Similarly, reusable, non-life supporting, non-life sustaining devices that are restored to their cleared or approved version or have publicly available labeling that describes these features. And so there's an and here. So it's either you've restored it to its approved condition, or it has labeling that's publicly available, and that the device, that labeling specifies that device lacks that FDA clearance or approval. So that is one of the other situations. Then we also have reusable, life-supporting, life-sustaining devices that have been restored to those cleared, approved version. Same situation, or they have that physical copy of that that labeling and publicly available, and specified that the FDA has not cleared or approved that device. And then the fourth is the IVDs, the intra vitro diagnostics that are available for less than 2 years from the EUA transition date or the expiration date, whichever is less. So those are some of the things that, some of the categories that the FDA has specified and called out. Now, what is the proposed timeline? That's what everybody cares the most about. Well, the draft guidance, they don't have an actual timeline. The FDA examples within that guidance, however, have an implementation date of July 1st. That's just a date that they threw out there, but it is something that's on the horizon. So we do need to be thinking about this. So what is that transition plan? So if one of those 4 categories maybe don't apply to you, or if you're in that that OR category and you want to be an approved cleared device, which certainly should be on your horizon if you want to continue that distribution, there are 3 phases that the FDA has outlined within that proposed timeline or the proposed transition plan. So phase 1 starts the day of the FDA's announcement. So as soon as they announce, we have a date that we're working towards. The FDA expects those manufacturers to start submitting those adverse event reports. Now, if you were under or distributing your product under an emergency use authorization, you should have been collecting those adverse event reports along the way. And now you're going to have to submit those. Additionally, if you plan to continue that distribution, those manufacturers that were distributing these products, you should now at least be preparing those regulatory submissions so that they can be submitted by phase 2. So phase 2, if we move to that, 90 days in, the FDA expects those manufacturers to comply with 21 CFR 806. So that section of the regulation is reports of corrections and removals. And then also 21 CFR Part 807, subpart B through D, this talks about how you should register your device and what devices actually need to be registered and on the different processes that those specific devices need to go through. So 90 days in, FDA expects those things to be happening. Then 6 months from that implementation date, that initial announcement, the FDA is going to terminate those EUA declarations. And manufacturers at that point have to comply with all applicable device regulations. So that's Part 820, which is the QSR, Quality System Regulation, which we're going to be talking about a little bit. And you may have to comply with some of the things that Thor is going to be talking about as well, if you weren't already. So we'll do a little bit more deep dive into those aspects. All right, let's go into the FDA's proposed QMSR. So this is one that is very recent. February 22nd, I believe, is when this proposed came out, but I'll give you a little bit of history as well. If you maybe this isn't on your radar. So the history of the quality system regulations, the 21 CFR Part 820. Now some people specify, well, 0.184 is the actual quality system regulation, but it's published in 1996. ISO 13485 was also published in 1996. And the industry ever since has been wondering, are we going to align? Are we going to come to an agreement on this? The FDA, for the most part, Part 820 has been largely untouched since 1996. ISO 13485, we're on our 3rd revision now. So there are some questions there. We'll get to that in just a little bit here. But the FDA announcement came out that they proposed that they plan to align the QSR to 13485. That is something they've been working towards, or at least talking about, since August of 2018. But February of this year is when they came out with this. The purpose was to harmonize and modernize the quality system regulation for those medical devices. Some people may be wondering, what's driving this? What's the purpose? The FDA, some people give the FDA a hard time, but they are striving for something of a global harmonization and a simplification of those regulations. That truly is their goal right now, provided that they are not reducing it to the point that it is actually reducing quality. But anything that is duplicative, the goal is to reduce those standards, to reduce the burden for the manufacturers themselves while still producing a quality product. Okay, so all that aside, what is the transition plan? What are we looking at here? As I mentioned, this is a proposed rule, so there are still comments open until May 24th. So if you have a burning desire that something not be the way it is, feel free to reach out there. The good news is that this is not, or it should not be, a big lift for companies who have been striving towards best practices in this industry already. So if you're already ISO 13485 compliant, you're likely in a very good spot. And especially if you've been marketing in the US, the biggest lift are for those companies that have a US only focus. And maybe you're unaware of 1345. I'll give you a few resources at the end here that will maybe help you get to that a little bit quicker. But several new sections for Part 820 are coming. So Section 820.7, this is simply an incorporation by reference. So what this is saying is they are incorporating ISO 1345 within Part 820 to say, this is going to be how we expect you to follow a quality management system. And those are going to be the regulations. They're incorporating ISO 1345 by reference. There's some questions come up. Okay, does that mean 2016 is going to be hard coded because they have referenced the actual standard? I mentioned earlier, ISO 1345 has been revised a few times. And while that might be a concern to some, the ISO, let me see here. I have a note here, the guy's names, because I wanted to get this right here for you. Peter Linders, chair of the committee that maintains the ISO 13485 standard. One of his lines that was my favorite, he said, maybe small updates or clarification to the 1345, but there will be no actual updates for the next 5 years. So that's the expectation anyway. So that we'll see how that goes, but the FDA would be evaluating those. So this would be to 2016 as of right now. Moving on to Section 820.10. So these are the requirements for the quality management system. The scope of design and development. So I see ISO 1345. If you put those next to each other, Part 820, the design and development section correlates to 7.3 in ISO 13.45. The scope is going to be the same, and Part 820 is going to maintain that scope. So specific Class 1 devices will have design controls requirements, all Class 2, all Class 3, that sort of thing. Section 820.15 is going to be a clarification of concepts. So product is planned or proposed to be included, and a few other things such as that, so that the standard can still be followed and maintained. So these are supposed to be, or the expectation is ISO 1345 and Part 820 are substantially equivalent, or substantially similar is the wording that is used. This clarification of concepts is to ensure that stays that way. Section 820.35 talks about the control of records, and this is interesting because it starts specifying more information and more detail for complaints and servicing. It also specifies a little bit more detail for UDI information. Now, UDI is already a requirement per other sections of the CFR regulations, but it's going to be living here as well with a little bit more detail to kind of bolster up what ISO 1345 requires. And then finally, there'll be a new section, Section 820.45. The FDA has seen a lot of recalls around device labeling and device packaging. So they're going to give some more detail around 820.45, around that in 820.45. So those are the new sections that are coming. Now, things to consider with the quality management system regulations, there is a free online version of ISO 1345. So if it is completely new to you, you do have the option to go online. You have that search functionality. You can check it all out. There's also a handbook, which they don't like us calling it a handbook, but that seems to be what people call it. It's officially the practical guide to 1345. We will be including these slides in an email to you, so you will have links in here. You can go check those out as well. So one of the things to consider is that there will be a 1-year waiting period, or at least that is the proposed waiting period right now. AdvaMed actually, in a meeting with the FDA, recommended that it be a 2-year waiting period. So it may be 1 to 2 years. We don't know just yet, but that's what the industry has requested. The proposed rule is also going to result in the replacement of the current quality system inspection technique, the QSIT, with a new approach that would be consistent with the new QMSR requirements. That being said, I'll skip down to my fourth bullet point because these kind of go hand in hand. The FDA doesn't plan to issue ISO 1345 certifications, so don't expect that. Also, ISO 1345 certified companies will not be exempt from FDA inspections. So it's likely going to be something similar to MDSAP, but take that with a grain of salt because, again, that's still being hammered out. And then finally, I already mentioned that the FDA is proposing to harmonize to a specific version of ISO 1345. Future revisions will be evaluated by the FDA before being incorporated. So with that, I will hand it over to Thor and let him deep dive into some of these biocompatibility updates.
Thank you so much. And honestly, it's funny we're talking about 1345 because I'll be taking that same kind of ideas and breadth and jumping into the changes we're seeing with 10993. On that, the interesting thing about this is some people may say, well, 10993-1 was just updated not too long ago. Why are we updating it again? And that's because we can't stop changing things. So we are going to be doing some 10993-1 updates, and that's what we're going to kind of talk through today. I do want to kind of press home that these are draft updates. We just barely talked about them and kind of still discussing them internally. So what I'm going to be talking today is very high level. It's not the details because the details will probably change. But that also means that we'll have to keep on looking and what's going to happen going forward. But I just want to keep everybody aware that these things are going to be changing. It seems like I'm not, oh, there it goes. Oh, maybe I wasn't too fast. Okay, so some big picture on what is biocompatibility, why we're doing it, kind of thought processes behind this. So the goals of the presentation today is to discuss these changes to 10993-1 that we're considering in the ISO committee. We're also going to talk about where we currently stand on the acceptance of these proposed changes. Where did the votes come out at with the new draft? And then kind of what we're going on next. What are the thought process going forward? So that's the whole thought process of the presentation going forward. So first off, what is biocompatibility? So biocompatibility, let's see if I go back, sorry, it's a little bit delayed on it. Let's see if I can go back one slide. I'll just kind of talk through biocomp real fast. So biocompatibility, when we look at biocompatibility, we're really looking at the safety of a medical device to the patient. And previously in 2016, we updated a lot of the concepts within 10993 and biocompatibility in general. So, we looked at taking the old 109 FDA guidance documents in 2018, we updated the ISO standard, and all these changes kind of made it more like risk-based approach, more like 1480, the standard, the 14971 management framework. And so when we look at these changes, we're trying to even do more of these types of approaches where we start to look at more of a risk-based approach. We're trying to take more of an evaluation technique. So the interesting thing here is we're dealing with, and most of you understand this, is when we're dealing with biocompatibility, we have how the FDA looks at biocompatibility, and then we have pretty much what the rest of the world looks like. So we do have some differences in Japan, we have some differences in China, and Health Canada in the EU and the FDA. So when we're writing these documents, we have to try to appease the evaluations of all these regulatory bodies, and trying to make 10993-1 kind of a unifying document for all of them. Can you imagine what would happen if we had a 10993-1 that the FDA didn't recognize? That would be very difficult for the industry to be able to understand what to do for biocompatibility, because the notified bodies, for example, would take 10993-1 as kind of a law. They would require you to fulfill the law of 10993-1, and if the FDA had something different, then really we'd have to do two types of evaluations, maybe two different sets of testing. It would be very difficult, and so we really are trying to get those regulatory bodies on the same page with 10993-1. I mean, it would be great if we could get all of 10993 recognized by everybody, but there are some of those either portions of those other standards or the standard itself that maybe the FDA won't recognize, but we can kind of work around that. But when it comes to 10993-1, it's imperative that we get consensus throughout the whole world on accepting it. So with that, when we start looking at what is changes, what the current document that's in draft, so this is in draft right now, and it came with significant changes to the current version of 2018. So the biggest changes that we're doing is we're doing even more of a risk-based approach. So 14971, we're basically taking that standard and putting it within the framework of 10993-1. So they're actually very good. We're going to try to make it very similar concepts so that we try to harmonize the standards for a risk-based approach. So as you remember, the last time we did this change, we tried to move it away from a checkbox mentality of just doing testing and actually look at the risk of the medical device and to the patient to understand what is needed to mitigate those risks. Well, we're trying to take that even further. Also, one of the good things about this new draft is we're actually trying to focus more on the 3 R's of animal testing. For those of you that don't know about the 3 R's, it's replaced, trying to find alternative to animals. This could be in vitro, this could be risk assessments, things like this. Or if we can't, then we try to either reduce the number of animals we use. Can we get the same information from a smaller subset of animals on testing or than we previously had? Or refine the experiments that carried out so that we help the animals as suffer as little as possible. So the good thing about this new standard is we are focusing more and more on the 3 R's and trying to utilize that going forward within the document. So when we start looking at a little bit more of the changes, there it goes. The current document, oh, I'm sorry, I went too fast. So I can get it going. Sorry, Etienne, I'm like flying through this. Okay. The other thing that we're changing with this document is putting more emphasis on creating what we call a biological evaluation plan, or a BEP, because we all love our acronyms in this industry, right? So we can't just call it biological evaluation plans. We have to get a shorter name. So we call it a BEP. This is something that we currently already do, but they're done differently. So different places will do different things with it. Some people will just kind of call out the device and its category and give a list of tests and say, that's my plan. When we're trying to do more of a risk-based approach, there needs to be more emphasis on the risk. And that's what basically a biological evaluation plan is. It's to understand and actually tell a story of the risk of the medical device, and then kind of mitigate that risk with a plan. So identify where those risks are, and then what are we going to do to mitigate those risks. One of the first things that I always run into when we're writing a BEP, and then we're trying to put a little bit more emphasis in this document, is appropriate description of that medical device. This goes to, actually, I can give a good example to this is with our acronyms. Sometimes we throw around acronyms because we just feel like everybody knows what they are because we use them every day, like BEP or SOP or whatever you want to say, right? And we just think that people know what that is just because we look at it as a second language. What we don't understand is that someone who's never maybe done an SOP before or written a biological evaluation plan, that just sails right over their head. We get this in finance a lot when people talk about EBITDA or COGS and things like that. They throw out those names, those acronyms, like everybody knows what it is, and somebody who's probably never done finance is just wide-eyed. This is actually exactly the same with a description of a medical device. I've seen engineers that have spent their lives engineering, designing these medical devices, and they just take that inherent knowledge that they've developed over time and the education experience, and they just think that people will know what they're talking about when they say what it is, or maybe how it's used. But man, getting really detailed, in fact, I even recommend sending an extra device to the FDA with your submission, things like that, to really get into the descriptive nature of your medical device, talking about how it's used, its materials, its diameters, really helping educate the scientists that might be trying to evaluate the risk, or the FDA reviewer who's maybe a biological, it could be biomechanical engineer, or it could be a toxicologist, right? Or whoever's looking at this may not have the same background as you do. So really being over descriptive of your medical device is something that we want to put in the standard. Then we want to actually spend more time describing what characterization is of that device, not just description, but characterization. That comes from chemistry, that might come from the physical form, materials. We want to kind of give more guidance on what that is. Also, we're concerned about chemical degradation. This is something that we don't normally talk about with medical devices, but over time, we can start to have degrading materials, components, different chemicals coming off of it, different physical degradation, like we talked about on the next bullet point, which is during the wear. These are things that are biocompatibility issues that in the past, we didn't spend a whole lot of time, not everybody spent a whole lot of time thinking about it. But if you have a device that does wear, we've seen issues in the clinical space with this type of issue. We want to be able to think through that and spell it out. We're also going to talk about device failures or breakages, things like that. And ultimately, we're going to put into the document guidance on how to conduct a BEP and what do you need to include into that BEP. If you wouldn't mind maybe advancing the slide for me, it might be just a little bit easier. I apologize. Awesome. Okay, so this is actually, I just want to spell out, BEPs are not new, right? So this is from the FDA's guidance document on how they interpret 10993. I would assume that eventually we would even see another updated version of this, because this came out in 2016 and we're already obviously kind of past a lot of concepts in here. But one concept in here that does apply to the BEP is this paragraph. And I love this paragraph because it kind of spells out how to write a BEP, right? So you look at the bold components there, you need to include the material components, manufacturing processes. This is essential to talk about the processing that goes into your device and the potential residuals from that processing. We're getting more and more information on how to do this, what we look for, things like that. And then ultimately the clinical use of your device. Those are the three inputs that you need to put in your formula for risk assessment, okay? So then based on those three inputs, you take all the risks from those inputs and you come up from it, look at it from a biocompatibility perspective, and then you identify a plan. And that plan is the BEP. So even though we're trying to get more and more information on how to develop a biological evaluation plan to help users develop their plans more effectively, this is a concept that's been around for a little while, but we're realizing we need to give more advice and more guidance. That being said, remember, now that we're putting into a standard, we're going to start having regulatory bodies like your notified bodies, for example, want to see these specific things we're looking for. So it does put a little bit more emphasis on your upfront work. The benefit to it, though, is the more work you put up front on your BEP, the less actual testing you need to do on the back end. So the more we understand those risks, the better we feel about them, the less actual mitigation we have to do. So it does take a little bit more upfront work, but hopefully that means less back end work for you in your company. Next slide, please. So with that, I just want to spend a couple of minutes on what we are talking about: physical and chemical information versus material characterization. This gets so confusing for people when they start hearing things like extractable leachables, chemical characterization, material characterization, physical chemical, like we're throwing around all these phrases, and sometimes it's confusing to people because they don't know which ones, what to do which. So with that, we're trying to define more what physical and chemical information is, and also what material information is, and also help you understand where extractable leachables can and cannot play in each of those. So that would be part of the new standard. Also, I put those in here for you guys to kind of think about, but that would be part of the standard as another addition. So next slide, please. So when you start looking at physical chemical information, there is multiple options. In fact, I would say that most of these should be looked at. So not so much options, but paths that you need to go down. The first one is something that people think is easy, supplier information. If I need to have physical chemical information, I'll just get it from my suppliers, put the pressure on them, and that way I don't have to worry about it when I get it. I have the material information, chemical information, and we can move on. There's a couple problems with that. First, remember that we're not just worried about the materials, we're also worried about the processing you do to it. So even though getting supplier information is important, it's not like you can just check box it and move on. You still have to evaluate what you're doing to those materials in your process. But two, we haven't been very successful in getting the suppliers to do the right type of information, provide the right type of information. So if you can do this, great. I actually would recommend it, but it's not something you can always guarantee, right? So the other thing that's actually more successful, though, is predicate testing. And so I get this all the time. It's like, okay, we have a device that we've manufactured. It has clinical experience. We have, things that we look for in the clinical space as far as like complaints and things like that. We did all the biocompatibility testing on it. All we're doing is changing a little bit of it, or maybe we're making the same device with a different intended use, things like that. This should all be part of your risk assessment. You'll all be part of your BEP, right? So materials and processes, like we talked about from the FDA. So if you have experience with materials and processes with the predicate device, especially if it's used very similarly, then all those can be put into your BEP. And then ultimately, how the device is used, if you can't get good supply information, if you don't have good predicate information, if it's a high-risk device, then that's where like more material characterization might come in. So that's where you might get some more like extractable leachables or other types of FTIR, things like that, depending on the use, to help you mitigate or understand and mitigate the risk of the medical device. Next slide, please. Okay, so the other thing we're adding is we're trying to get more into what we call biological equivalence. And this goes back to that predicate concept, where if your composition, physical form, processes is the same as another approved medical device, then why are we requiring further testing and characterization, right? So if we're able to demonstrate that similarities between an already device on the market with those things we talked about, those inputs, the materials, the physical form, residuals, things like that, then we should be able to, get away from doing further characterization and data sets like testing. Next slide, please. So that will be in the new standards, some more guidance on that. And actually, like I said, that should help us when we're starting to evaluate, your risk of a medical device. I always tell companies that you should start really developing a database of biocompatibility on your products. The larger companies do this already, but like if we're worried about materials and processes and physical form, if those are things we're worried about, we can start a database of materials and processes and things like that with testing done on it. So when you're looking at developing a new medical device or changing a medical device, you can go to your database and understand what risks you know and what risks you don't know to help you guide on the testing needed. This is super, once again, it takes work up front to develop a database, maintain it, things like that, but it saves you so much time on the back end if you do it correctly. One of the more controversial parts of the new draft, and yeah, this slide's fine, thank you, that we're still evaluating the need and kind of what it looks like is actually getting some individual endpoint guidance within -1. So traditionally, -1's been kind of the umbrella general document, and then things like cytotox or sensitization, irritation, hemocomp have been done with their individual portions of the standard, like -5, -10, -23, -4. What this new draft's kind of done is we still have those standards to give in-depth guidance. But the thought process is how many people go to -1 for general, just have -1 for general guidance. And so with -1, maybe we should have appendix or other information, just high-level information about guidance on evaluating cytotoxicity or subacute subchronic toxicity, implantation effects. So when you start looking at it in the standard itself, there's been a little bit of work done to kind of give high-level guidance on how to do some hemocompatibility evaluations or how to do degradation or toxicokinetic studies. I'm not sure if they're going to stay in there because, like I said, it's one of those things where it's like we do have our own standards to evaluate these. And so this could be repetitive work, but some of the drafting group does feel like it's beneficial to have some high-level guidance in -1 since maybe someone's just using -1 only, or -1 kind of tells you to do a biological evaluation plan and evaluate these risks, but don't give you any information about those risks themselves and just refer to the individual standards. So that is something that we're still evaluating. I'm not saying it's going to go away or stay, but like I said, something that's in the draft right now. Next slide, please. Before we go on this one, I do just want to remind everyone, if you have any questions about these presentations, please put them in the chat. That's, to me, always the more valuable part. I get tired of hearing my own voice, so I'd love to kind of hear from you guys, and then we'd love to answer any questions you guys have. So we did recently vote on 1093-1, the new draft. It did pass, so it is technically the new working draft document. There was a negative vote from both US and Germany on the document due to some of the issues we kind of talked about, maybe some disagreements that usually happens within the standard. Obviously, having the US be a negative vote is impactful, because that is a big portion of, not the only portion by any stretch, but a good portion of the US vote does come from the US FDA. And I can tell you the US FDA does have some issues with the draft itself. So like I mentioned before, this is something we're very concerned about. We want to make sure that everyone can agree on 1093-1. It's one of those that we probably can't just say, okay, well, the FDA don't accept it, or have your own guidance. This is something we probably need the whole world to kind of be on board with. So we did meet to discuss the issues with the draft as a working group. There are a lot of rework ideas, so there's a lot of comments on the draft, as you can expect with 2 negative votes. And I think it was just a really good working session. I think the spirit of the draft is good. The things that we're talking about, more risk-based approach, less animal testing when needed, things like that, I think everyone's on board with. I think just the details that get kind of, have to be talked about with something that's this extensively changed. And something that wasn't that old to begin with, right? So we've made some significant changes in the last 5 or 6 years. So I think it's just details that we need to work through. So we met, we talked about those issues, went through the comments. Both US and Germany made presentations on why they or we thought that the document needed to be worked on. In that one, the decision was to kind of continue with the current draft. So it wasn't to revert back to the old one and work on that one. It was to continue with the current draft that was proposed, and then form different writing teams to work on the problem areas that the negative votes had and the comments were. So that's where we're at right now. We're working on the working groups of different individual teams working on different areas of the -1 draft. And the concept there would be that we would come back with new verbiage, new suggestions, things like that, and submit that again as a draft to get more votes. So that's kind of the work. But I kind of wanted the next few slides to work through something that we're working on. One of the things that was discussed heavily, and one of the things that the concepts that's going on. So next slide, please. So one area we're working on is the chart. So everyone knows that 1093, the 2018 version that we released, had big changes to this chart. So we added, in fact, go ahead and click one more time. I have it highlighted. We made some changes to this chart in the draft. So we had physical chemical information that we added as a column. This is a new column that wasn't there before, requiring, as you can see, an X to do physical and chemical information. Next slide, or next touch, please. And then, okay, that was a mistake. That was left blank. So that's sensitization, but as you can see, that's what there was cytotoxin irritation. Next one, please. And then we added material-mediated pyrogenicity, which before was in just acute systemic toxicity. So it's a whole new column itself, even though before we kind of had to do it anyway. Next, go ahead and click it one more time. And then we separated subacute and subchronic toxicity and tried to identify when those would be needed based on the use of the device. So before it was just subacute subchronic, and you decided when it was needed. We try to give more guidance there. And I think one more click, please. And then carcinogenicity. This is something previously before chemical characterization, we kind of always had, but no one ever did because the testing is so expensive and long. But now with chemistry, extractable leachables, if you're required to evaluate this endpoint, then there's a way to do that with ENL, with extractable leachables. So that's something that now the FDA specifically really focuses on good extractable leachables to be able to identify carcinogenicity concerns. So go ahead and click one more time. And then last, we added those reproductive toxicity and degradation endpoints at the end. As you can see, nothing was marked to evaluate. It's kind of those things where if you think you have one of those concerns, you need to do it. Okay, so next slide, please. So this was the draft. This is the slide we did in 2018. I'm going to talk through some concepts of what the new kind of thought process may be. So I've kind of transferred that slide into something that looks worse, but I'll tell you why it looks worse in a second. So this is the transitioned over. The concepts here is, if you click the next slide, please. If you look at how some patterns in here. So first off, we have what we call cytotoxicity, sensitization, irritation. We call that the big three. At least I call that the big three. And everything's required of that. Okay, so we need to evaluate cytotoxin, sensitization, and irritation for every device, regardless of contact time or duration. Next slide. Another thing to think about, though, is you start to see other patterns within this chart. So here I've kind of made this stepping looking pattern where you start looking at how different contact types, regardless of the contact type and duration, you start having the same pattern. In fact, next slide, please. As you can see, this pattern is kind of repeated as we go down. So the thought process when we start looking at this is the chart. The whole concept of 2018 was to get people away from checkboxing, right? So to get people away from, and that's where we put material/chemical information as the only thing that's required. Everything else was an E. But we're also starting to see once you have a table, people are naturally going to checkboxes. It's just your engineering brain, for those of us that have engineering brains. Oh, table, checkbox. It's hard to get away from that thought process. So one concept is if we start to see these reoccurring patterns in the table, can we change it from a table to more of a flow chart? So next slide, please. Oh, we have one more on this one. Go ahead and click it one more time for me. The next, oh, sorry, go back. There is nothing over on reproductive and carcinogenicity. So it's kind of like we have that up there, but we don't tell any guidance. So let's make this into a flow chart. So go ahead and next slide. Okay, so are we going to do a new flow chart? Let's take a look at what this new flow chart looks like. So the first one, and I'm sorry, those things aren't showing up. So I'll go ahead and just work through it here in a second. It's in the next slide, so go ahead and do the next slide. So the first one is the, well, let me work through the, in fact, is it okay if I share my, can you give me a presentation?
Yeah.
That might be,
yeah,
sorry
absolutely.
about that. I'll make sure this is corrected when I send it out for people.
Let's see.
Yeah, if you want to make me a presenter. Perfect. And tell me, oops.
I see a castle.
You see a castle. Okay. Do you see my deck yet? How about yeah?
There it is.
Okay. So this is the new proposed flow chart. Oops, I'll just do it from this side so we can kind of go from here. So the new flow chart here is evaluating for the big three. Like I mentioned before in this previous deck, if you look at that orange color, everything was the big three. So we're going to evaluate for the big three. Next is if it has mucosal contact, then you need to look at systemic toxicity. If it contacts greater than 24 hours, then you look at implants. If it's greater than 30 days, you evaluate for genotox. If it's not mucosal, but if it's breached or compromised surface with indirect blood, then you need to evaluate for material-mediated pyrogenicity. If that contacts greater than 30 days, then you evaluate for genotox, carcinogenicity, and also the findings of that genotoxicity to see how the carcinogenicity may work. And then if you do need to do genotoxicity, maybe you do chemical characterization to look at those other hazards like reproductive toxicity. If you have direct tissue contact, you need to evaluate for genotoxicity plus the other things. So this builds on one another, right? So if you have that, you do the previous boxes, and you need to look at if it's greater than 24 hours genotox. And if you have direct or indirect blood contact, then you need to evaluate the hemocompatibility endpoints. If it's circulatory system, you look for implant studies there. If it's, you need to look at genotoxicity, if it's extracorporeal. So you need to look at hemocompatibility, but you build on the other ones. So this is just one proposed work that we work offering for the new chart. Like I said, the concept there is that everybody has a hard time getting away from checkboxing a chart. So if we can get it away from there, that's better. So where do we go from here? Like I mentioned previously, our main goal is to have 10993 that's recognized by the FDA, but it still incorporates more of a risk-based approach, more emphasis on in vitro alternatives. A good example of that is our new ISO 10993-23, right? So -23 is a standard for irritation. It used to be with -10. So -10 used to be sensitization and irritation. We've broken -20 irritation out to -23, and the emphasis in -23 is the in vitro alternative to the rabbit irritation test. We're still working through some of those acceptance from the FDA. The rest of the world's accepted -23 and the in vitro test. But we're trying to help -1 put more emphasis on in vitro alternatives. Can we evaluate the same endpoints without using animals? And if we can, then we should probably rely more on the in vitros, as long as we've proven to be competent in vitro alternatives, and less on the animals test. Plus, we want to do more alignment with 14971. We want to have those two ISO standards kind of mirror each other, because we want it more of a risk-based approach. So that's the main goal. We want to be able to do those three areas, but still have the FDA recognized -1. So that's what the writing groups are working on right now. So with that, I know we have about 13 minutes left. I want to turn it back over to you, I think, because there's another slide for you. I apologize.
Oh, yeah, okay. I'm glad you mentioned that. Let me take back over. While I'm doing that, one of the questions is there a red line going to be available, and how does that work as far as seeing the previous to new?
Yeah, there will be events. So for those of you that are not aware of the procedure that's going to go through, right? So right now, the different working groups within like AME or Belgium or UK have looked at the draft already. They're the ones that made comments and voted. So we'll go back through and incorporate those comments like I talked about, try to rewrite it. We'll send it back out to those, to the countries' groups to get re-voted on. As soon as we actually get more of a finalized draft document and get it into an actual document, there will be a red line potential. And honestly, you'll have to do a gap. So as soon as 1093-1 is approved, the new one, there'll have to be a gap analysis to the old one for your current testing. So one of the things that was mentioned before about things that have the emergency use authorization that are getting pulled, if you need biocompatibility done on that device now, and you wait for some reason until this new -1 is approved, you will be under the new -1, right? So depending on your device, that could be a good thing. If it's a low-risk device that has a predicate, then maybe you want this new -1 because it will help you. I can tell you that these concepts in this new -1 aren't necessarily new. We use them all the time when we write BEPs, and we're just trying to give more guidance in that. So, but there will be a red line, yes. That's a long answer to that question. I apologize.
No, that was great. And another follow-up question to that, I guess, would be the timeline. As far as you mentioned, the new verbiage, new draft that might be coming in hopes that the U.S. might be on board. Any thoughts on that?
Yeah, if I was good at this, then I'd be much richer in the stock market. Anybody who's ever worked in the ISO document space knows that this can take a while. It just really depends on how well we address the comments. You know, there's thoughts that a new draft could be happen later this year, like in the summertime. You know, spin that out for new comments and go back. Maybe 2023 is beginning of 2023 is a possibility. But like I said, there's a lot of changes to this draft, and so it's one of those things where I want to be careful saying that.
Yeah. You mentioned the database of testing that you recommend setting up. Are there any more best practices around how to do that or what you've seen?
Yeah, and so it depends on how many devices your company manufactures and how similar they are, right? So if you're an orthopedic company and you make a lot of stainless steel, titanium, cobalt, chrome types of devices going through very similar types of processing, it makes a lot of sense, right? Because what you can do then is say that we have people even send in coupons that represent different types of processing for those materials, that would replicate the surface characteristics and the process residuals. And that's how they build their database. Other people wait for it based on just if they need to test and it's new, and then they collect that information. Really, though, you need to be able to tell the story with it. So you just can't say, this device was tested before, it's the same material, I'm good. You really need to be able to tell the story of why you feel like it's appropriate from a processing standpoint. But it's a lot easier for manufacturers who have very similar types of use in materials, like orthopedic devices. For those of you that make a broad range of devices that use a bunch of different polymers, it becomes more different, because even if you have a polypropylene from a supplier A and you have a different polypropylene from supplier B, those aren't comparable. They have different additives, they have different things in the mix. So that makes it harder to justify. So those kind of things you might want to think about when you're building it on exactly the type of information you put in. But it's so useful to have to be able to justify how to biocompatibility test if you do it right.
Yeah, okay, good answer. I like the detail. One of the other questions that came to mind were the 3 R's that are changing, or a more focus on the 3 R's. I wondered if you had any examples as far as how, I mean, you mentioned one at the end, turning that table into a flow chart and maybe using certain in vitro alternatives. Can you expound on any of that, or maybe add some examples that you've seen?
Yeah, so one that we're doing right now that's successful is getting away from the in vivo thrombogenicity test, or the dog thrombogenicity test. For those of you that know, it's a horrible test. It really is. Not even just because it uses canines, which I think most people don't love, but secondly, the test itself is not the most predictive, consistent test. And so give FDA a lot of credit here. They've done a lot of work on understanding the faults in the test. They brought in experts to present to them, and they've very aggressively looked for alternatives to that test. And so there's different loop methods or in vitro test tube methods that if you submit the protocol to the FDA on a pre-sub and say, this is how we're going to look at in vitro or thrombogenicity, along with in vitro alternatives that are traditionally there, like hemocompatibility, PTT, PLC, things like that. Those are acronyms again that we use, platelet and leukocyte counts and partial thrombosity, things like that. Then we can use those in vitro alternatives in that suite of in vitro tests to justify out of the animal tests. And that's something we do right now. So we haven't ran an animal test in a very long time. Also, a lot of these in hemocompatibility, or sorry, vascular types of devices, they do functionality tests before they go to biocompatibility to see how it performs. So they'll use large animals to run to make sure it functions correctly. If we can know that beforehand, we can help put in hemocompatibility endpoints into the functionality test. And the FDA loves that too, because it's done, once again, in an animal, but we get more use out of that animal than just functionality. We get safety too. The other one I mentioned was the in vitro alternative. So -23 is out. The in vitro alternative has been validated. We've done tons of round robin work. We've published a lot of studies comparing that to animal data. Everywhere in the world accepts that in vitro irritation test. It's done on reconstructed human epidermis skins. I have other presentations that actually go into details if people are interested. But the FDA right now is the only one that's kind of holding up universal acceptance of that. And we're working through, we have a group of people in the industry working through the FDA. It's called the MDDT process, where it's a kind of an innovative technology that you go through the FDA for acceptance. We're working through those questions now. We've been working through a little bit, but the COVID kind of hurt us as far as availability for that. But now we're getting back on track, and hopefully we can get the FDA to buy into that one. So that's another animal test that we can replace. But next would be sensitization. And then so we're just going to try to kind of really cut down on these animal tests to a point where hopefully my goal is to get to a point where I don't have to do animals unless I'm doing like an implant, things like that.
Awesome. Well, that's exciting to hear. Sometimes we kind of get a little scared when we hear about these changes to the regulations and standards, but I think that's definitely a huge plus in a lot of people's minds, definitely mine. So we have about 5 minutes left. I don't have any other questions. I don't see any other questions coming through. Let's give everybody a few minutes back in their day. Thank you, Thor. I enjoyed it. I'll just remind those watching, if you want to reach out to Thor or myself, information's on the screen. It will also be emailed to you after this. So let us know if you have any questions or feedback. We'd love to hear it. Any last words, Thor?
Now, thank you for everyone attending. It's always fun to talk about, and look forward to continuing to keep you updated on changes to the new standard.
All right, we'll let you get back to it. Thanks. We'll see you all later.
Bye-bye.